Good day, everybody. I hope you're doing well. I hope you're, kind of going with the class now and kind of figuring everything out. And remember, you can always, email me with any questions you have. I'll usually try to get back to those emails within 24 hours. So, as long as you're ahead of time and not waiting till last minute, I pretty much can answer all the questions for you. Um, Today's video is going to, or videos, is going to focus on chapter 3, where we are going to be going over the structure and function of cells. Um, with this module, we're going to start off with the first video discussing kind of background information on cells, their kind of general components, and then we're going to focus on animal cells, which are just like human cells, and kind of the organelles and the different structures and their functions for those general cells. Then after that, in our second video, we're going to look at diffusion, osmosis, active transport, and passive transport, and kind of how molecules and ions move in and out of the cell, whether or not they require energy to do so, or if they can do so passively. And then the final video for this module will talk about and discuss how our cells make energy, ATP, out of glucose. So we'll be focusing on glycolysis and cellular respiration, and the organelle that's responsible for kind of guiding all of that, which is the mitochondria. And so that's the videos that we're going to have in this module. So all of that's going to focus on information from chapter 3. And the first video, we're going to get into the structure of cells and all of the different organelles that general animal cells have. All right, so the first thing we're going to look at is the cell doctrine. And so basically the cell doctrine just kind of or gives off the importance of cells and organisms, living organisms. And basically what the cell doctrine says is that all living things, so anything classified as living, is composed of cells. And that kind of harps back on chapter 1, where we talked about the characteristics of living organisms. That was kind of one of the stipulations, is that they have to be made up of cells, and there's some organization to that cellular makeup. The second thing, or second trait dealing with the cell doctrine, is that the single cell is the smallest unit of biological organization that is classified as living. So it has all the characteristics of life. And if you remember, with biological organization, there are the atom, molecules, and macromolecules before the presence of cells in that biological organization. Those first kind of 3 levels that we've talked about does not have all of the characteristics of life. But when you get to the level of the cell in biological organization, that's when you see the characteristics of life. So all of those characteristics of life. So we can say that the smallest level of biological organization that is living is the cellular level. Okay? And the third part of the cell doctrine says that all cells come only from pre-existing cells. And so those cells have to divide and replicate themselves to make daughter cells that will, then go on and continue to replicate and divide. And we'll talk all about that when we learn about mitosis and meiosis much later in the session. That's like one of the final chapters that we'll go over. Okay, so the cell doctrine basically states these important characteristics of cells and how they're kind of related to life and to living things. Now, if we look at the general characteristics of cells, we have prokaryotic cells and eukaryotic cells. And remember that prokaryotic cells are in the bacteria domain and the archaea domain. Okay, and then eukaryotic cells are in the domain eukaryote. Okay. And now remember, these are more of like your single-celled, more simplistic, no membrane-bound organelles. All right, so no membrane-bound organelles, whereas eukaryotic cells are mostly, right, most of them are multicellular. There are some protozoans that are unicellular, but a majority of the percentage of eukaryotic organisms are multicellular. They do have membrane-bound organelles. So I'm just going to say membrane organelles, and they tend to be more complex, right? And larger, right? And whenever we look at some of the general characteristics between these prokaryotic and eukaryotic cells, so if we get more specific than these generalities, we see that both cells have a plasma membrane. Right? That plasma membrane, if you remember when we talked about lipids or phospholipids, is what kind of separates the internal portion of the cell from the external area. So you have that intracellular area and the extracellular area, and that plasma membrane is responsible for that separation. And as we'll learn later on, the plasma membrane is really important for dictating and governing the material that flows into and out of the cell. So it's very selective in terms of what enters and leaves the cell. Now, with prokaryotic cells, we said there's no membrane-bound organelles, and so that means there's no nucleus. Right? There's no other organelles or structures inside of the cell that has a membrane. The only membrane in prokaryotic cells is that plasma membrane. And then there's a fluid that's within the cells, so inside of that membrane, so that intercellular fluid, that's the cytoplasm. And you see that in both the eukaryotes and the prokaryotes. The prokaryotic cells, while they have no nucleus, the eukaryotic cells, they do have a nucleus. So they have that membrane-bound nucleus. And they also have other membrane-bound structures that we'll talk about in this video. And each of those structures have a specialized function. So they have a specialized job and role that they perform within the eukaryotic cell. Now, prokaryotic cells, they don't have those true membrane-bound organelles. One of the kind of only overlapping organelles between prokaryotic cells and eukaryotic cells are ribosomes. Right? So those ribosomes, they're the organelles that make the proteins, and so prokaryotic cells and eukaryotic cells both have proteins, so they need those ribosomes in order to function and to have those proteins to be made in order to carry on those cellular processes and cellular functions. And so we've also mentioned bacterial cells are prokaryotic, and then an example of eukaryotic cells are human cells. So we have a microscopic image here, a micrograph of a eukaryotic cell on the left, prokaryotic cell on the right. And what you can see is you have the plasma membrane. So in the prokaryotic cell, it's kind of right there, okay? Whereas the plasma membrane is this outer area here on the eukaryotic cell. This outer area here, this kind of dark green line there, is not the membrane for the prokaryotic cell, but that's a cell wall. So prokaryotes have a cell wall, while animal cells do not have a cell wall. But another cell that does have a cell wall are plant cells. Right? So plant cells also have a cell wall. You also have cytoplasm kind of in this area. You have cytoplasm in this area. Here you have the nucleus, which is this purple blob here, and it's bound by a membrane. And that's where the genetic material is found. In a prokaryotic cell, that genetic material is not kind of concentrated in a membrane, it just kind of floats around inside that cell. And you can see the light green material, and that's the genetic material that's floating around in the prokaryotic cell. And then all of these other structures, these round structures that you see in this image on the left in the eukaryotic cell, all of these structures, right, are other membrane-bound organelles. So all of those are organelles that, again, have a specific function and a specific role to carry out in those eukaryotic cells. Now, even though eukaryotic cells are really similar, right? So we have this generality that we say eukaryotic cells have in terms of their characteristics. But so even though they're really similar, there are a lot of structural differences within eukaryotic cells. Right? So if you think about it, all the cells in our body are eukaryotic cells. And all of our cells don't look the same. They don't have the same function. They don't have the same role in our body. So for example, you have muscle cells, which have many organelles, especially mitochondria, right, which is responsible for, I'll just put an arrow here, for the energy. And so they have a lot of organelle, or a lot of mitochondria for that reason. And they also have a lot of nuclei. They're multinucleated. So you can have multiple nuclei in one say skeletal muscle cell. Compare that to nerve cells. Nerve cells are long and thin, and a nerve cell could be, several inches long compared to a muscle cell that's going to be relatively small. And that's because that nerve cell is used to carry impulses, action potentials, as we'll learn, over a long distance, say up our spinal cord, or from our fingertip to our spinal cord, or vice versa. And what's really important is the small size of cells. So cells being small actually provides an advantage of being really efficient in terms of moving material in and out of those cells. And we'll look at why that's the case here in a few slides. Now, just to show you some examples of differences within these cell types, you've got muscle cells on the left here. So this is, I believe, a skeletal. No, this is cardiac muscle. It's right here in the heart. You can kind of see the intercalated disc there. And so each of these little lines have is a muscle cell. So this area is a muscle cell. And another way you could tell it's cardiac is it's not multinucleated. Skeletal muscles are. And so each of these are their own individual muscle cell. That looks much different from this individual nerve cell, which also looks very different from this individual cell that's in the tubule of a kidney that's responsible for helping filter our blood of waste and to produce urine. So very different structures, very different functions, even though they all are eukaryotic cells that have the same organelles and kind of the same general composition. And like I mentioned in the previous slide, cells are small because that allows an increased efficiency in terms of moving material in and out of the cell and across and throughout the body. So if you have a box or a cell, let's say our cells are small, right? Being small means that they have a very high surface to volume ratio. And I'll show you an example of what we mean by that in the next slide, okay? But just know that small cells have a high surface to volume ratio than larger cells. So basically, if you're smaller, you have more surface than space inside of your cell. So let's imagine the cell as a box or a cube. If your box is large, the surface on the outside of the box is small compared to the large space inside. And so if a cell is small, that surface on the outside is actually larger than the space on the inside. And this matters because the high surface to volume ratio allows cells to diffuse material and to move material in and out of the cell much more efficiently. So they're much more efficient at acquiring nutrients and getting rid of waste through active and passive transport mechanisms because they are smaller. So it's much more efficient at moving things across that cellular membrane. So here we have an example where we have one large cell versus 8 small cells that's covering the same amount of volume. Okay. Now notice if this cell has an ion in the center here, if it wants to travel out of the cell, it has to move this distance, right? Whereas if the ion is at this point in the cell, that distance it has to travel, right, is much smaller compared to the larger cell. So that space inside, that volume is smaller compared to the surface area. So this example has a higher surface area to volume ratio. Okay, so surface area to volume ratio. And our cells can be even more complex and increase that surface area to volume ratio by having these finger-like projections kind of protrude from the surface of the cell. And the space in this projection there's not a lot of volume in there, but because this projection is extended vertically, it's kind of stretched out. The surface area to volume is very high. So the nutrients and the waste don't have to travel very far to diffuse out of that cell. And as a matter of fact, our digestive system has microvilli on the surface of the cells because it increases absorption. And what's really interesting is if you zoom into those finger-like projections, so let's say if we drew one, there's even more finger-like projections on that villi, right? So there's folds upon folds of that, and all that is leading to an increased surface area to volume ratio, so that they can be more efficient at diffusing and moving material in and out of the cell. Okay, so that's a very important concept in terms of cellular efficiency. All right, so now that we've talked about kind of the general characteristics of eukaryotic cells and prokaryotic cells, we're now going to look at the organelles and structures that we can find in a eukaryotic cell. So this is an image of a typical animal cell where you have all of the organelles shown here. And we know it's an animal cell based off of organelles that are present and organelles that aren't present. So if this was a plant cell, it would have chloroplast, and it would also have a cell wall outside of the plasma membrane. And because it doesn't have chloroplast, because it doesn't have a vacuole, and because it doesn't have a cell wall, we know that this is an animal cell. And so some key organelles that are going to be in eukaryotic cells are the plasma membrane. Where's that labeled at on here? Am I blind? I believe I am. Anyway, it's this structure here. So this orange layer, right? That's our plasma membrane, okay? Now, inside the cell, our organelles, we're going to start at the center and kind of work our way out. Our organelles, first the nucleus, which is this purple structure. And the nucleus is where the DNA is contained. And the nucleus is where ribosomes are made as well. Okay. And then attached to the nucleus is what's called the endoplasmic reticulum. So this blue membranous structure, these blue folds, are two types of endoplasmic reticulum. The one that's directly attached to the nucleus is the rough endoplasmic reticulum. And the rough endoplasmic reticulum gets its name because it has ribosomes attached to the outside of the surface of the endoplasmic reticulum. And if you remember, I said those ribosomes are really important for making proteins. And so the rough endoplasmic reticulum, one of its primary jobs is to make proteins. And so the ER, the rough ER, is the primary site for protein synthesis in a eukaryotic cell. Now, when you move away from the rough ER, you get to the smooth ER, where the smooth ER is primarily responsible for other organic macromolecule synthesis, so like lipids, those sorts of macromolecules. And so they're not primarily making those proteins because they don't have the ribosomes that are needed to make those proteins. And then once you leave the smooth ER, you get to this pancake-like membrane structure that's called the Golgi apparatus. The Golgi apparatus is basically like a package area where it ships those molecular products either inside the cell or out of the cell. And it ships those products via vesicles, which are these round kind of spheres. And you have many different types of vesicles, which we'll talk about more specifically, but this one's showing a secretory vesicle where it's pushing and releasing substances from the inside of the cell to the outside of the cell. So it's basically secreting that material. These little rod-like structures that you see here, the purple ones and the microfilaments, those are referred to as the cytoskeleton. And there are different types of cytoskeleton components, but they're really the structural framework of the cell. They're how organelles kind of move around the cell, and they basically provide that framework. You can think of it as like the studs in the walls inside of your house. There you have the centrioles, these yellow structures. These are really important in cellular division. We'll talk a lot about centrioles when we talk about mitosis and meiosis. Centrioles are what kind of grabs onto the DNA and helps to separate the DNA as it divides. Here we have another vesicle, which is a peroxisome. This is used for mainly getting rid of waste. Here you have a lysosome, which is another vesicle that's used for breaking down old organelles that don't function properly anymore, or any kind of cellular debris or foreign invaders that come into the cell. And then the last one that we need to mention is the mitochondria. The mitochondria, like we mentioned, is what produces the energy for the cell. So it takes a molecule of glucose, and under a really efficient process, it can make approximately 36 molecules of ATP. And so that's where that happens. That cellular respiration happens in the mitochondria. Okay, and so we're going to go through kind of this pathway in a little more detail, going from the nucleus to the Golgi apparatus and out of the cell, and then kind of talking about the structural components as well. Another thing I need to mention is this light yellow color that's all inside this cell, with that the organelles are kind of floating in. That's the cytosol. So this is kind of a gel-like material that's inside the cell and kind of keeps everything in place along with the cytoskeleton. Okay, and so the first thing we're going to kind of focus in on is the nucleus. Okay, so the nucleus, like we mentioned, is where the genetic material is found and contained, and the nucleus basically controls all of the activities of the cell. So you can think of it as the control center or the hub or city hall of the cell. I tend to think of the cell parts as pieces of a city. And so I think of the nucleus as like city hall, the governing building, the governing structure inside of the city. And so that's going to tell everything else what to do based off of the proteins that are made from the RNA that's going to leave the cell, which basically comes down back to DNA. And we'll learn all about that in a later chapter. Now, some characteristics of the nucleus is that it's actually a double layered membrane. And we got an image on the next slide that we're going to show. So there's actually two membrane layers that's kind of keeping the DNA enclosed. But the DNA, like we mentioned in our chemistry lecture, the DNA is converted into RNA, and remember that RNA is converted into proteins. Right? Well, DNA is in the nucleus, right? And RNA, if it's made from DNA, is in the nucleus, and somehow it has to get out of the nucleus in order to interact with the ribosomes so that it can be made into proteins. And the way that the RNA leaves the nucleus is through these nuclear pores. Okay? And now the DNA exists in the nucleus as chromosomes or chromatin. And so these terms can be kind of confusing. So chromatin is basically uncondensed DNA. So the DNA is in its long strands. You can't see it under a microscope, whereas chromosomes are duplicated, condensed DNA that you can see under a microscope. And so that's kind of the distinction that I want you to have right now. But as we talk about DNA in a later chapter, we'll really go in and make a distinction between these two. And then the last portion, or the last part that's in the nucleus, is the center area, which is the nucleolus. And the nucleolus is where the ribosomes are made. Okay? And then the ribosomes will be made, and then they'll exit through the nuclear pores. So here's an image. So we got a micrograph using an electron transmission microscope. So this is a 6,000 times magnification. And this purple area here is the nucleolus. It's at the center of the cell. And if we zoom in on like a cross section, you can see the double membrane here, and then also the nuclear pores that the RNA is going to exit so that it can go into the cytosome, not the cytosome, but the cytoplasm, and interact with proteins. Not interact, man, I can't talk. The RNA will leave the nucleus through the nuclear pores into the cytoplasm to interact with the ribosome to make proteins. There we go. I think I said that right. All right, and so like I mentioned, the ribosomes are responsible for protein synthesis. Okay? And so we said that the ribosomes are found on the surface of the endoplasmic reticulum, and that's referred to as the rough endoplasmic reticulum. One thing I forgot to mention when we were looking at the cell image and all the organelles is that there are also some free-floating ribosomes. So if you notice, there are these little orange circles here, right? Those are free-floating ribosomes that are present in the cytosol, so they're not attached to the surface of the endoplasmic reticulum. Now, the difference between these ribosomes is that the free-floating ribosomes typically make proteins that are going to be used immediately in the cell, whereas the proteins that are made in the rough ER are most likely going to be transported and exported out of the cell, okay? And so you can think of your endoplasmic reticulum and your ribosomes as kind of these manufacturing plants for different products of the cells, right? And those products are proteins, lipids, those types of macromolecules that we've talked about. And like we mentioned in that figure for the cell, the ER is this network of membranes that's folded around one another. And again, that increases surface area, having all of those folds. And so we have those two types of endoplasmic reticulum, the rough again, which has the ribosomes that are going to make proteins to be shipped out of the cell. And then you have the smooth ER that has no ribosomes on the surface. And again, this is for some of those other macromolecules like fats and lipids. But it also will package proteins and the lipids that it makes so that they can be delivered to the Golgi apparatus. Okay? So you can think of the ER as kind of the manufacturing plant within the city. And then once they make their products, they then move those products to the shipping plant, which could be, our mail carriers or Amazon, right? Where those products will be shipped to wherever they need to go. And the place that ships those products is the Golgi apparatus. Okay? And now, again, if we look at that image of our cell, we have the nucleus, you have the rough ER in this area, and then you have the smooth ER. So rough ER is proteins, smooth ER is lipids, hormones, those types of macromolecules. And then once those products are made, they're then shipped. So you can see this little sphere here. That is a vesicle that has basically budded off of that endoplasmic reticulum. And it has products inside, and that vesicle will merge, like you see here, with the Golgi apparatus, and it will transfer those products into the Golgi apparatus. Now, when the products get to the Golgi apparatus, they basically refine, repackage that material so that it can be shipped out to the various places that it needs to go, whether it's inside the cell or it's outside of the cell. And so again, you can think of the Golgi apparatus as a packaging and shipping center, so something similar to like an Amazon. And the products, just like with the endoplasmic reticulum, are going to be put into vesicles that will bud off of the membrane of the Golgi apparatus, and it either will go to other locations in the cell or it will be exported out of the cell. Okay. All right, so let's just look at the figure again, just to kind of remind us. Right, so the Golgi apparatus is, I think of these stacks of pancakes. So that's this structure, that green structure there. You can again see the ER vesicles budding to or attaching to it, releasing products. And then the products will kind of travel through the Golgi this way. And then the membranes will bud off, carrying the products either into the cell or out of the cell. So either through a kind of like a transport vesicle, which is what that would be, or the secretory vesicle, which is what you see here with those products being released out of the cell. Okay. This is often referred to as the cis side and the trans side of the Golgi. So cis is the one that's closer to the interior. Trans is the side that's closest to the exterior of the cell, and it's basically the kind of the receiving side and the shipping side of the Golgi apparatus. And now, kind of following our discussion of the different vesicles, right? So we were saying, we have like a transport vesicle, you have a secretory vesicle. There are other vesicles as well. We've talked about secretory, so that's containing products that's going to be exported from the cell. We have endocytic vesicles, which brings substances into the cell. So this is a form of exocytosis, and then this is endocytosis. I will show that in a little bit. But basically, out of the cell and then in the cell. Okay. Peroxisomes, like we mentioned earlier, get rid of waste that are produced by the cell. So under cellular, as the cell's completing different cellular processes, it's generating waste and byproducts, and we have to get rid of those byproducts in our cells. And peroxisomes are what does that. So they have enzymes that will break down those wastes and make them non-harmful to the cell in our bodies. And then we have the lysosomes, which have the digestive enzymes that are breaking down like foreign invaders, like a bacterium, or old organelles that are broken down and not working anymore. And so they will break those organelles down into their individual components so that they can be recycled and reused in new organelles. So here we have images of these four types of vesicles. So you have your lysosome. This one is taking up a bacterium that it's going to break down, and then it basically turns into a secretory vesicle where it ships out that material. Here you have a peroxisome that is taking up cell waste, breaking it down, and then turning into a secretory vesicle to release those harmless wastes out into the extracellular environment. And then the blood can pick up those wastes and take it to our kidneys or our liver, depending on the type of waste it is, and it can clean it out of our body. All right, so the next organelle we're going to look at is the mitochondria. And so the mitochondria is the power plant, PG&E of our cells. And depending on the cell type, the number of mitochondria is going to differ. So for example, muscle cells have a lot of mitochondria, and cells that aren't responsible for generating a lot of energy don't have a lot of mitochondria. And mitochondria is interesting because, like the nucleus, is also surrounded by a double membrane. The inner membrane is really folded in on itself. It has a ton of different folds. It kind of looks like an endoplasmic reticulum on the inside. And that inner membrane is where the electron transport system is of the cellular respiration process. And again, we'll talk more about that in the third video for this module. But basically, cellular respiration takes oxygen and produces CO2 while also taking glucose and making ATP. And so all this happens in the mitochondria. And so here we have an image of a mitochondria. So we have a micrograph. You can see the folds on the inside of that inner membrane. What I want you to notice is there's a space between the outer membrane and the inner membrane, and then there's the space on the inside of the inner membrane. Now, those are going to come into play. Those spaces are going to be important whenever we talk about cellular respiration. So just keep that in mind. Keep the structure of the mitochondria in mind, because we're going to come back to that. And so you can see different products going into the mitochondria that's going to be responsible for helping to facilitate cellular respiration. And through the process of cellular respiration, you get out ATP and carbon dioxide. Okay. And just to mention again some sources of energy, we've already talked about, or I've already mentioned glucose. Glucose is stored in animals as glycogen. So glycogen, if you remember from our organic lecture, the chemistry lecture, is just a long molecule of glucose. And so glycogen is a carbohydrate. And this glycogen is basically a short-term energy storage in animals, and we store that in our muscle cells and liver cells. And our carbohydrate stores are generally the first kind of energy sources that we use whenever our body needs to break down molecules to obtain ATP. And so that's the glucose. Now, if you're low on glucose, if you're low on carbohydrates, your body can tap into the long-term energy stores, which are in the form of triglycerides, or those lipids, those fatty acids that are in the adipose tissue. And so we have two kind of sources of energy. Now, proteins can also be broken down and used for energy, but our bodies tend to do that as a last resort because our proteins are so important in kind of regulating our cellular processes that we really don't want to be breaking down proteins in order to make energy. Cells also have structures that enable support and help maintain the typical structure and shape of the cell itself. And then cells also have structures that are used for movement. The cytoskeleton, like we mentioned, is important for support. So it's basically like the internal walls of the house, the internal scaffolding, and that helps to maintain the cell shape. Now, cells can also move via a couple different structures. So cells can have cilia, flagella, or centrioles. And we'll kind of look at some images of those. But cilia are a lot of little finger-like projections that help the cell move. Flagella are usually single, longer microtubules that kind of beat back and forth like a tail that helps to propel it. And then the centrioles are structures that kind of turn in order to provide kind of a propulsion. Again, we have some figures that are going to hopefully give you a better idea of what those are. Here are just kind of some more details about the cytoskeleton. So there are two different components that are part of the cytoskeleton. You have microtubules and microfilaments. So just remember that hollow is a tube, right? So that's the microtubules. And then the solid fibers are the microfilaments. Both of these are made up of protein, okay? And they both make up that framework, that internal scaffolding, kind of the structural support of the cell. And they also serve as an anchor for other organelles, and organelles can also kind of use those to move around the cell. And so here we have an image where we're showing this is a microtubule, because you can see it's hollow, and then you have the microfilaments that are not hollow kind of running off those tubules, and then you can see they're attached to the mitochondria, the Golgi apparatus, kind of holding everything in place, and that they also attach directly to the plasma membrane via these proteins that span the length of that membrane. So those proteins that are in the membrane serve as kind of an attachment site for those cytoskeleton components. And so for the movement part, the cilia, like I mentioned, are short, little finger-like projections, and there's usually a lot of those. And one of the more common places to find cilia is in our airways, so our respiratory tract. And so those finger-like projections actually help capture, say, foreign objects that enter our respiratory system, so like dust, dirt particles, those sorts of things. They get trapped in the cilia. There's also a mucus there, and then that's why we have like the mucus buildup in our noses and in our respiratory tracts, and that helps to push those products back up and out of our bodies. The flagella is usually long and single. There can be double or I think even triple, but those are less common. And so that flagella is just basically like a long tail that helps to allow that cell to move. And so one of the more common examples of a cell with flagella is a sperm cell, so the spermatozoa. It uses that flagella to swim into the fallopian tubes to fertilize the egg during fertilization. fertilization. And even though they're slightly different kind of structurally in the way they look, internally, cilia and flagella are really similar in terms of how those, not projectiles, but how those projections move back and forth to cause that movement. Now, the last one, centrioles, are these rod-like microtubule structures that are important for cell division. And so what the centrioles do is they kind of reach out and grow and attach to the chromosomes that are duplicated, the duplicated chromosomes in a cell, and they pull those duplicated chromosomes apart, and they allow for that DNA to separate into the different daughter cells. And so they're moving the DNA um, to the opposite ends of the cell. And if it wasn't for the centrioles kind of pulling that DNA to the other side, you wouldn't always get equal amounts of DNA in the daughter cells during mitosis or meiosis. Okay. So here's an image of cilia. Right, so all of these finger-like projections are cilia. Here you have a bunch of different sperm cells with their long flagella, and then the acrosomes, the head of the sperm cell. And if you take a cross section of the flagella, you can see these microtubule filaments that are kind of connected to each other, and they just shorten and lengthen at different times that cause kind of the back and forth movement of the flagellum on the sperm tail. Okay. One of the last things we're going to talk about in terms of cell structure and characteristics is the plasma membrane. And like we mentioned earlier, the plasma membrane is really important for determining the flow of material in and out of the cell. It separates the internal portion of the cell from the external portion of the cell of the cell, which is that extracellular environment. And it's what we call selectively permeable. So it can select what moves in and across the cell. And so it's permitting movement of some things, but not others. And that's allowing specific information to be transferred in and out of the cell between the environment and the cell itself, and also between other cells. And so the plasma membrane has a really, really important function in every cell. And like we mentioned in our organic lecture, plasma membrane is a lipid bilayer. So you remember that lipid bilayer has the glycerol head with the two fatty acid tails, the phospholipid. And so it has that polar head, right? So that polar head and then the non-polar tails. And so remember the non-polar tails do not like water, whereas the polar head does like water. And so remember that causes them to orient in a certain way to create that plasma membrane. Now, proteins are also embedded in between those phospholipid molecules. And these proteins provide a means of transport for molecules through the membrane, because sometimes there's a polar molecule that wants to move in or out of the cell. And because the phospholipid has a non-polar tail, those polar molecules can't make it through, or they may be too big to make it between the spaces of the phospholipids. And so it needs that protein in order to serve as a channel to allow those molecules to basically move through. You also have cholesterol that is embedded within the plasma membrane, and this gives some rigidity to the plasma membrane. But again, at the same time, the plasma membrane is also very fluid and dynamic, and it's kind of this free-flowing, free-moving membrane. And because of that fluidity, it's known as a fluid mosaic. So the fluid coming from that kind of moving fluids component mosaic, given the fact that it's made up of all of these different components. So phospholipids, cholesterol, proteins, and then the final thing we haven't mentioned, which is carbohydrates. And so carbohydrates will be attached to the proteins, and they serve as this communication molecule between the cell and other molecules, or the cell and adjacent cells. And so that plasma membrane, like I mentioned, is non-rigid and is very fluid. And so it's almost kind of like this floating fluid dynamic structure. So here we have an image of a typical plasma membrane where you have your phospholipid bilayer, which is highlighted by this rectangle here. You have different types of proteins and channels that allow different ions and molecules to move in and out of the cell. We'll talk about those a little more later. Then you have these cholesterol molecules, right? Remember the 4-carbon ring, cholesterol molecules. And then you have this carbohydrate that is attached to the protein. And again, that's that communication with the cell, with its external environment. Okay. We have a little video here that kind of goes through the plasma membrane again.
The plasma membrane is the outer boundary of an animal cell. It's made up of lipids, proteins, and carbohydrates. Phospholipids are the major components of the plasma membrane. These molecules have hydrophilic, water-loving heads and hydrophobic, water-fearing tails. The phospholipids form a double layer called the lipid bilayer at the surface of the cell. The hydrophilic heads on one side interact with the extracellular fluid, and on the other side interact with the cytoplasm inside the cell. Interspersed in the phospholipid bilayer are proteins. Some proteins are embedded in the membrane and span the bilayer. Others are located on one side or the other, often attached to membrane-spanning proteins. Molecules of cholesterol are also scattered throughout the bilayer. Carbohydrates attach only to the outer surface of the plasma membrane. Most of the carbohydrates are attached to proteins, forming glycoproteins. Glycoproteins often function in cell recognition. The structure of the plasma membrane is often described as a fluid mosaic. The proteins are interspersed among the lipid molecules like tiles of different colors within a mosaic. Many of the proteins are able to move sideways through the bilayer to some degree, giving the membrane its fluid quality.
All right. And so that video is just really good at kind of showing you how, or what exactly we mean by fluid membrane and how the molecules are kind of moving amongst one another. Even though it's held in place, they are still kind of free floating and moving, which is pretty interesting. All right, so that does it for the first video in chapter 3, where we went over general characteristics of cells and then the organelles and their structure and function that we typically find in animal cells. What we're going to look at in the next video is how ions and molecules move in and out of the cell across the plasma membrane via passive transport and active transport, and how osmosis, diffusion, and this term called tonicity plays a role in the movement of all of those substances. So with that, we'll see you in the second video.